Good morning, everyone. It is 10 a.m. This is Dr. Amanda Cohn, the Executive Secretary of the Advisory Committee for Immunization Practices. I am going to go ahead and turn it over to Dr. Romero to bang his virtual gavel and open the meeting.
Good morning, everyone. Welcome to the August 26th, 2020 ACIP meeting. Thank you for giving up of your time. This is another one of our virtual meetings open to the public. So with that, I just knocked on the table. If you couldn't hear that, this meeting is now open. Back to Dr. Cohn.
Thank you, Dr. Romero. So good morning and welcome to the August 2020 virtual ACIP meeting. Copies of the slides that are being presented at today's meeting are available until 5 p.m. today on the ACIP website. These slides will be removed from the website this afternoon, made 508 compliant, and re-put up on the website in the next week or two. Additionally, slides are available through a share file link for ACIP voting, liaison, and ex officio members. Next slide. Next slide. Slides presented at this meeting will be posted on the ACIP website in the next couple of weeks. Additionally, the live website webcast videos will also be posted on the ACIP website approximately 4 weeks after this meeting, and minute meetings are posted on the ACIP website generally within 120 days of the meeting. We have not yet posted minutes from the July ACIP meeting, but the videos are available. Next slide. This is for the ACIP voting ex officio and liaison members who are on the Zoom call. Please mute your lines at all times until you're called on for discussion. When Dr. Romero opens the meeting for discussion, please virtually raise your hand. And during the discussion period, Dr. Romero will take questions first from the voting ACIP members and then from ex officio members and liaison members. We are going to stay on schedule per the meeting agenda. So if we're running early, we will break until the next start time on the agenda. And we ask that everyone on the Zoom open call, please disable your video. Next slide. I want to call everyone's attention to the next two ACIP virtual meeting dates. These remain tentative in that meetings may be added, but we can assume that the next meeting as of now is on September 22nd, 2020, starting at 10 a.m. and running likely until 4 p.m. Eastern Standard Time. And we will be having a meeting on October 28th and 29th, 2020. This is our normally scheduled ACIP meeting. And during that meeting, we will be able to discuss topics that are not COVID related. Next slide. This meeting's oral and written public comment processes are designed to accommodate increased public interest in ACIP's work, maximize opportunities for comment, make public comment more transparent and efficient, and create a fair process for assigning limited oral public comment time. Next slide. Members of the ACIP agree to forego participation in certain activities related to vaccines during their tenure on the committee. ACIP has issued limited conflict of interest waivers to members who conduct clinical vaccine trials to serve on data safety monitoring boards. These members are prohibited from participating in committee votes related to those vaccines. And regarding other vaccines of the concerned company, a member may participate in discussions with the provision that he or she abstains on all votes related to the vaccines of that company. At the beginning of each meeting, ACIP members state any conflicts of interest. At this meeting today, there are no votes scheduled So while ACIP members will be stating their conflicts of interest at the beginning of the meeting, there will be no vote occurring where they would have to, where they would be prohibited from being part of the meeting. Next slide. So I'm now going to hand it back over to Dr. Romero, who's going to start with roll call. I'm going to shift the direction slightly from the last time we did roll call. For ACIP members, when Dr. Romero calls you, please state your name and conflict of interest only. Ex officio and liaison members do not need to state their conflict of interest or affiliation. Dr. Romero will call the organization, and the member who is representing that organization, please state your name. Dr. Romero?
Thank you, Dr. Cohn. I'll begin. Jose Romero, no conflicts of interest. Dr. Atmar?
Robert Atmar. I'm a co-director of the Clinical Operations Unit of the NIH-funded Infectious Diseases Clinical Research Consortium that is working with the COVID-19 Prevention Network to evaluate SARS-CoV-2 vaccine candidates in phase 3 clinical trials, including the vaccine produced by Moderna that will be discussed in today's meeting. Over.
Thank you. Dr. Alt?
Kevin Alt, no conflict.
Ms. Bata?
Lynn Bata, no conflict.
Dr. Bell?
Bell, no conflict.
Dr. Bernstein?
Frank Bernstein, no conflict.
Dr. Fry?
Hello, I am we have a VTEU site here at St. Louis University. That's a vaccine treatment evaluation unit, which is part of the Infectious Diseases Consortium for Research, Clinical Research, and I will be conducting COVID vaccine trials.
Thank you. Dr. Hunter?
Paul Hunter, I as well amount of Pfizer stock and a very small grant for Pfizer to do quality improvement project related to pneumococcal vaccines. Thanks.
Dr. Lee?
Grace Lee, no conflicts.
Ms. McNally?
Veronica McNally.
Any conflicts, Ms. McNally?
No conflict.
Thank you. Dr. Paling? Dr. Paling?
Kathy Paling, no conflict.
Thank you. Dr. Sanchez?
Pablo Sanchez, grant from Merck for antimicrobial vaccine.
Thank you. Dr. Szilagyi?
Peter Szilagyi, no conflicts.
Dr. Talbot?
Kip Talbot, no conflict.
That ends the roll call of the ACIP members. I will now begin the roll call for ex officio representatives.
Before we start the roll call for ex officio members, we are hearing a lot of echo on the line. Can I please ask for anybody who is not speaking to mute your line? Thank you.
I'll begin with ex officio representatives. Centers for Medicare, Medicaid Services.
Mary Beth Hansen.
Thank you. Department of Defense.
Brian Schumacher.
Thank you. Department of Veterans Affairs.
Jane Kim.
Thank you. Food and Drug Administration.
Lauren Fink.
Thank you. Health Resources Services Administration.
Mayor Rubin.
Thank you. Indian Health Service.
Thomas Weiser.
Thanks. Thank you. Office of Infectious Diseases and HIV Aid Policy. Office of Infectious Disease and HIV Aid Policy.
Tammy Beckham.
Thank you. National Institutes of Health.
John Bible.
Thank you. I'll now move on to liaison representatives. Again, state your name only, no need for stating conflict of interest. American Academy of Family Physicians.
Pamela Rockwell.
American Academy of Pediatrics.
Yvonne Maldonado.
American Academy of Physicians Assistants.
Marie-Michelle Léger.
American College of Health Association.
Sharon McMullen.
American College of Midwives.
Carol Hayes.
American College of Obstetricians and Gynecologists. American College of Obstetricians and Gynecologists. I'll come back to them. American College of Physicians.
Jason Goldman.
American Geriatric Society.
Ken Spader.
American Immunization Registry Association.
Rebecca Coyle.
American Medical Association.
Sandra Freithofer, American Medical Association.
American Nurses Association.
Chad Riddle.
American Osteopathic Association. American Osteopathic Association. I'll come back. I'm sorry. Thank you. American Pharmacists Association.
Steve Foster.
Association of Immunization Managers.
Chris Finley.
Association for Prevention, Teaching, and Research.
Paul McKinney.
Association of State and Territorial Health Officers. Association of State and Territorial Health Officials, forgive me. I'll come back. Biotechnology Industry Organization.
Phyllis Arthur.
Council of State and Territorial Epidemiologists.
Christine Hahn.
Canadian National Advisory Committee on Immunization. Canadian National Advisory Committee on Immunization. I'll return. Infectious Diseases Society of America.
Carol Baker.
International Society for Travel Medicine.
Elizabeth Barnett.
National Association of County and City Health Officials.
Matt Zahn.
National Association of Pediatric Nurse Practitioners. National Association of Pediatric Nurse Practitioners.
Patsy Stinchfield.
National Foundation for Infectious Disease.
William Schaffner.
National Medical Association.
Pat Whitley Williams.
Pediatric Infectious Diseases Society.
Mark Sawyer.
Pharmaceutical Research and Manufacturers of America.
Corey Robertson.
Society for Adolescent Health and Medicine. Society for Adolescent Health and Medicine.
Amy Middleman.
Yes, thank you. Sorry. Society for Healthcare Epidemiology of America.
Marcy Drees.
Okay. Let me go back around. American College of Obstetricians and Gynecologists. American Osteopathic Association. Association of State and Territorial Health Officials. That completes the roll call. Thank you very much. I will turn it back over to Dr. Cohn.
Great, thank you, everybody. We did forget to introduce Dr. Nancy Messonnier, who is here as well, representing the Centers for Disease Control and Prevention. We know that we are having some echoes that are coming from the room, and we are working on that as we speak. We are going to mute everyone on the phone except for the first speaker. And we apologize for the inconvenience, but we are working on it. Thank you. Dr. Romero, are you ready to move to the first part of the agenda?
I am. So we're going to talk about coronavirus disease in 2019, vaccines, with an introduction by Dr. Bell. Dr. Bell, please.
Thank you, Dr. Romero, and welcome to everyone. There are the slides. Thank you. I'm hoping that I'm not echoing as I'm speaking. It will be hard to understand me if I am. Could I have the next slide, please? Thank you. Over 200 COVID-19 vaccines are currently under development, including 4 in clinical trials in the United States at the moment. The ACIP has been responding to this ongoing pandemic and accelerated vaccine development through scheduling of monthly emergency ACIP meetings, during which we have the opportunity to discuss evolving epidemiology and other relevant data and hear about the vaccines as they're being developed. At the July 29th meeting, to remind all of you, we reviewed the status of COVID-19 vaccine clinical trials. We discussed COVID-19 vaccine safety considerations. We spent some time considering the differences between FDA licensure versus emergency use authorization of COVID-19 vaccines. We discussed vaccine implementation. We reviewed the epidemiology of COVID-19 in essential workers, including healthcare personnel. We discussed our ongoing work on COVID-19 vaccine prioritization and discussed how the work group was approaching these issues. And we reviewed the evidence to recommendations framework. Next. Our COVID-19 vaccine work group meets weekly, and we have been meeting weekly since the last ACIP meeting. During August, we covered a review of COVID-19 epidemiology among at-risk groups, including American Indian, Alaska Native populations and individuals with underlying medical conditions. We heard from manufacturers and discussed the clinical development program for 2 mRNA vaccines, including data from phase 1/2 clinical trials and plans for phase 3 clinical trials. We'll be hearing from these manufacturers today. We discussed models that have been developed that model allocation strategies to inform initial COVID-19 vaccine supply. We reviewed initial COVID-19 vaccine distribution scenarios, and we continued our work focusing on vaccine prioritization considerations, especially for initial doses. Next. On today's agenda, we will be hearing from clinical development of mRNA 1273 by Dr. Jacqueline Miller from Moderna. We will be hearing about the Pfizer BioNTech COVID-19 mRNA vaccine clinical development from Dr. Nicholas Kitchen. We will discuss an overview of post-marketing safety surveillance from Dr. Tom Shimabukuro. We will hear from Dr. Nancy McClung from CDC about the epidemiology of COVID-19 among individuals at increased risk of COVID-19 disease. We will hear from Dr. Rachel Slayton about modeling allocation strategies for the initial COVID-19 vaccine supply. A work group, sort of the work group interpretation of our current situation, and prioritization and work group next steps from Dr. Kathleen Dooling. Next, please. I wanted to just spend one more moment talking about considerations for prioritization of COVID-19 vaccines. As I think as more information has become available, the work group has been able to advance our thinking on this topic with a view towards fulfilling our obligation to issue some guidance and recommendations. So in today's session, we will focus on individuals at increased risk of COVID-19 disease, including underlying medical conditions and age. And then we will review the 4 priority groups for consideration of allocation of initial COVID-19 vaccine. We will now have heard in some detail about each of these 4 groups at ACIP meetings and, of course, also in the work group. In the September session, our plans are to focus on the epidemiology of risk of COVID-19 by race and ethnicity, to review other published prioritization frameworks that may have been completed by that time. And we will be looking to a possible vote on an interim prioritization schema for allocation of initial COVID-19 vaccine when it's available in limited doses. Next, please. So I always like to take this opportunity to show the work group members, including the ACIP members, the ex officio members, many liaisons, and the CDC co-leads to whom we are in debt, as well as our consultants who are absolutely pivotal to our work. And next, here's a list of all the CDC participants who help us understand the data as it becomes available. And I think that's the end of my presentation. I want to thank you all for joining and look forward to a very productive meeting.
Thank you very much, Dr. Bell. So let me ask Dr. Miller from Moderna to please present mRNA 1273 clinical development. Dr. Miller, please.
Thank you, Dr. Oh, apologies. Thank you very much. My name is Jacqueline Miller, and I am the therapeutic area head for infectious diseases at Moderna. And on behalf of Moderna, I'd really like to thank the ACIP and the working group for the opportunity to give you this update on our phase 1 clinical data and phase 2, phase 3 clinical trial plan. Next slide, please. This slide is the standard forward-looking statements and disclaimer slide. Next slide, please. So mRNA 1273 is a vaccine intended for protection against the SARS-CoV-2 virus. It comprises a messenger RNA sequence, single sequence, against the spike protein. And that protein has 2 proline substitutions, and therefore the protein is called S-2P. Those 2 proline substitutions keep the messenger, the translated protein in the pre-fusion conformation, which is the most immunogenic form. The messenger RNA is delivered via lipid nanoparticle targeting the antigen-presenting cells, including dendritic cells and monocytes. Once the messenger RNA is delivered, the ribosome translates that messenger RNA into the SARS-CoV-2 protein. It naturally assembles into its wild-type trimeric conformation and then is cell surface expressed. The vaccine is a 100 microgram dose, and I will describe some of the dose ranging data in the slides that follow. In addition, the vaccine is a 2-dose schedule given at 28 days apart. The vaccine is shipped and stored at -20 degrees, and then is able to be stored at the point of care at 2 to 8 degrees in the refrigerator. Next slide, please. So first, I'd like to give you an overall summary of the preclinical data that enabled us to progress forward with clinical development. In collaboration with the Vaccine Research Center at the NIH, a robust preclinical data package has been compiled in mice and non-human primates. In both species, robust neutralizing antibodies have been observed to be induced, and these neutralizing antibodies led to protection against lung challenge in the mice and pulmonary and nasal challenge in non-human primates. In addition, after challenge, there's no indication of enhanced respiratory disease on lung pathology after viral challenge, even when lower and subprotective doses of messenger RNA 1273 are used, giving us reassurance into going into clinical development regarding vaccine-enhanced respiratory disease. And finally, a Th1 dominant phenotype of CD4 cells has been induced in both mice and NHPs. So with these data, we had discussed with FDA progressing into our clinical development program, which is detailed on the next slide, please. So this is a program that has advanced relatively quickly over the last 7 months or so. And it's really the messenger RNA platform technology that has enabled that, along with frequent reviews of data with the FDA. Our first time in human study, which has been sponsored by the NIH, was initiated in March and continues to be ongoing. And the data I will be reviewing for you today come from both published data last July in adults 18 to 55-year-olds, as well as older adult cohorts who are 56 to 70 years of age and above 71 years of age. Then I'll describe to you the phase 2 and phase 3 study designs, both of which are funded by our partners at BARDA. The phase 2 study initiated in May and enrollment is complete, and the phase 3 study started on July 27th. Additional considerations for the clinical development program in the future would be in pediatric populations, women in immunocompromised populations. Next slide. So now I'd like to describe to you the study design for the phase 1 clinical trial that I'll describe in the subsequent slides. So again, this is an NIH-sponsored phase 1 safety and dose ranging study that was conducted in 3 different age cohorts, so 18 to 55, 56 to 70, and greater than 71 years of age. The initial plan was to evaluate 3 dose levels in all 3 age strata, so 25 microgram, 100 microgram, and 250 microgram in a sequential manner with stops for safety in between. A fever to 39.6 degrees Celsius was observed in a recipient 18 to 55 years of age with the 250 microgram dose. And therefore, the 25 and 100 microgram doses were the ones continued into the 2 subsequent older age cohorts. And then finally, 3 additional cohorts have been added to evaluate the vaccine at 50 micrograms. This study is currently fully enrolled and in its follow-up phase. So these subjects were primarily healthy males and females above 18 years of age, and we enrolled all comers with respect to their baseline SARS-CoV-2 serostatus, although subjects that described a past medical history of COVID-19 disease were excluded. Safety endpoints include adverse reactions for 7 days after each injection, as well as unsolicited adverse events within 20 days after each vaccination. Serious adverse events and medically attended adverse events will be followed through the entire duration of the study. And the immunogenicity assays that are being evaluated in this study include the ELISA, pseudoneutralization assay, that is the primary assessment for neutralizing antibodies moving forward in the development plan, live virus neutralization, and an intracellular cytokine staining assay for CD4 T cells. And again, these subjects will be followed for approximately 1 year after their final vaccination. Next slide, please. So I described to you that the 250 microgram dose was interrupted due to a single fever observed in a participant 18 to 55 years of age. Now I'd like to describe for you why the 100 microgram dose has been selected for further clinical development. What I'm showing you on this slide are the neutralization antibody titers with the pseudovirus neutralization assay However, the conclusions are similar with the other assays investigated. So what you see in the graph moving left to right are the 25 microgram, 100 microgram, and 250 microgram doses in the subjects 18 to 55 years of age. Highlighted in pink are the middle two quartiles of a range of convalescent sera. So these are 41 subjects who were tested after presenting with symptoms of COVID-19 disease. And what you can see from the slide is as of day 14 post-dose 2, neutralizing antibodies were observed in all participants at all dose levels. However, the lowest responses were seen at the 25 microgram dose. For the subjects in the 100 and 250 microgram dose groups, the range of antibody titers was in the upper half of range of convalescent sera. And so the higher antibody titers observed with the 100 microgram dose versus the 25 microgram dose was the reason for selection of that dose moving forward. Next slide, please. So this slide now depicts the safety data with the 100 microgram dose in all three of the age strata, 18 to 55, 56 to 70, and greater than 71 years of age. And on the bar graphs, you see in gray the grade 1 reactions, grade 2 are in blue, and grade 3 are in orange. The vaccine was well tolerated across age groups with injection site pain and then the solicited symptoms of fatigue, chills, headache, and myalgia being the most frequently reported symptoms. There were more reports observed after the second dose of vaccination, but there was no increase in reactogenicity observed in the older age cohort. The symptoms were primarily mild to moderate in severity, and the majority of these symptoms resolved within 2 days. Next slide, please. So now I'd like to take you through the immunogenicity data for the 3 different age strata. And now on this slide, what you see are binding antibodies that have been measured by an ELISA assay across the 3 age groups. And again, 18 to 55 at the left, 56 to 70 in the middle, and greater than 71 years of age in the right. Again, highlighted in pink are the middle 2 quartiles of the range of convalescent sera. So with this 2-dose series, 100 micrograms seroconverted all subjects after the first dose of vaccine. And the area under the curve also exceeded the median of the convalescent sera. After the second dose, all age groups are equivalent to the higher range of titers of the convalescent sera in the upper quartile of the dose. And importantly, these results are consistent regardless of age stratum. Next slide, please. So now on this slide, you see, again, the 18 to 55, 56 to 70, and over 71 years of age presented in the pseudo-neutralization assay. So these are the neutralizing antibody titers. And once again, after the second dose, we note pseudo-neutralization antibody titers in all participants, including the upper age quartile, or upper age range. The titers were comparable across the 3 age strata, and I've included the geometric mean titers in order for you to be able to observe the titers achieved. And in all age groups, the median titer was above the median of the convalescent sera at day 57 post-dose 2. So that's 1 month post-dose 2. Next slide, please. So finally, I'd like to review the CD4 positive T cell data that were generated in the 3 age strata in the phase 1 study. So this is looking at the CD4 T cell response, and it's looking at the Th1 phenotype as measured by elaboration of interferon gamma, IL-2, and TNF alpha. And similar to the humoral immunity assays, the CD4 Th1 T cells were detected across the 3 age strata. The Th2 phenotype was rare, and the data are not shown in the slide, but they can be seen in the Jackson publication for the 18 to 55. And so with that, I'd like to really thank the NIH for allowing us to present these data, in particular the data in the older age cohorts that have not yet been published. Next slide, please. So now I'm going to shift gears and give you an overview of our ongoing clinical development program. We have 2 additional studies ongoing. The first is a phase 2 study that is funded by BARDA and sponsored by Moderna, which is evaluating the safety and immunogenicity of 2 dose levels, 50 micrograms and 100 micrograms. We've enrolled 2 cohorts, the first cohort 18 to less than 55 years of age, and then a second cohort greater than 55 years of age. Both of those cohorts include 300 subjects, and amongst those 300 subjects, 100 subjects each will receive 50 micrograms, 100 micrograms, or placebo at day 1 and day 29. Again, these are healthy males and females, and we have enrolled all comers with respect to baseline SARS-CoV-2 status. The safety endpoints are comparable to the ones I described for the phase 1 study, and we will be investigating the ELISA assay and the neutralization antibodies as generated by the pseudovirus neutralization assay in this phase 2 study. The follow-up will be approximately 1 year after the second dose for all subjects, and this study is now fully enrolled. Dose 2 has been administered to the entire population, and the subjects are undergoing their active vaccination follow-up. Next slide, please. So now I'd like to talk about COVE study, which stands for the COVID-19 Efficacy and Safety Study. This is our pivotal phase 3 efficacy, safety, and immunogenicity study, which will ultimately enroll 30,000 subjects. This is a study that is conducted through multiple partnerships, including BARDA funding and the partnership of NIAID and the COVID-19 Prevention Network. It is a phase 3 study that will investigate the safety, efficacy, immunogenicity in subjects who are either under 65 years and not at risk, and that will comprise approximately 60 to 75% of the population, those over 65 years of age, and combined with a cohort younger than 65 years of age who are at increased risk for complications of COVID-19. So these 2 strata, the older age cohort and those younger subjects with comorbid conditions, will comprise at least 25% and up to 40% of the study population. And so this was really designed in mind with generating results that are generalizable to the U.S. population that is at risk for COVID-19 disease. The study started on July 27th, and as of Friday last week, there were over 13,000 subjects enrolled. So subjects will be randomized 1-to-1 to receive either 100 micrograms of vaccine or placebo. And again, these subjects are all comers with respect to their baseline SARS-CoV-2 serostatus. The study has 2 co-primary objectives. The first is to demonstrate the efficacy of mRNA 1273 to prevent COVID-19 disease, and I will be providing you the case definition in the slide that follows. And the second is to further evaluate the safety and reactogenicity profile of the 2 dose regimen of mRNA 1273. This study will last for approximately 2 years after the second vaccination for each subject, and the intent is for full safety and efficacy follow-up. Next slide, please. So the case definition to define COVID-19 disease for the primary analysis in the COVE study is that the participants must have experienced either 2 of the listed systemic symptoms, fever, chills, myalgia, headaches, sore throats, or new smell and taste disorders, or 1 respiratory symptom, cough, shortness of breath, difficulty breathing, or clinical or radiographical evidence of pneumonia. And the participant must also have at least 1 nasopharyngeal swab, nasal swab, or saliva sample positive for SARS-CoV-2 by RT-PCR. And the preference is for the nasopharyngeal swab taken in the clinic, but some allowance has been made for subjects who may be too sick to travel to the clinic. The primary analysis will be conducted in seronegative subjects, so those who are initially SARS-CoV-2 baseline serostatus negative, and cases begin to accrue at day 14 after dose 2, aligned with the immunogenicity data I presented for the phase 1 study. Next slide, please. So I would like to take a moment to talk about an initiative that I'm quite proud of at Moderna. So we have brought together a Diversity and Inclusion Advisory Committee. And the purpose of this committee is to help us achieve our goal to enroll a study population that is representative of the racial demography in the U.S. We are committed also to transparency in this initiative, and if you are interested in weekly updates for enrollment as well as current minority enrollment status, you can find those at the Moderna webpage if you click on the COVE study. So currently, the study has enrolled 18% minority, and with the help of our Diversity and Inclusion Advisory Committee, we hope to improve upon those numbers. This committee is helping us review enrollment, race, and ethnicity, helping us review and evaluate outreach activities, helping give advice on strategies so we ensure participation of individuals from communities who are deeply impacted by COVID-19, and supporting the development and implementation of retention strategies in the trial for these subjects. And so thanks goes out to all of our collaborators on this committee. Next slide, please. So finally, a note on the limitations of research. While the data I have shown you are very encouraging in terms of the immunogenicity of the vaccine, they are at the moment limited and in a fairly homogeneous population. We also need to think about how we will evaluate the vaccine in pediatric subjects, pregnant women, and immunocompromised patients. But the ongoing COVE study will provide significantly more data, both in healthy adult subjects as well as in older age cohorts and subjects with pre-existing comorbidities. Next slide, please. So in summary, the messenger RNA 1273 vaccine encodes the pre-fusion stabilized spike protein with the 2 proline substitutions in a lipid nanoparticle dispersion, which is designed for delivery to the antigen-presenting cells of the lymph node. Our preclinical data package has demonstrated induction of neutralizing antibodies and protection against viral challenge in mice and non-human primates. The interim data from the phase 1 study indicate that a 100 microgram dose of vaccine is generally well tolerated across the age strata, with solicited symptoms mostly mild to moderate in severity and self-limited in duration. The vaccine has been observed to induce neutralizing antibody in the upper half of the range of convalescent serum across age strata, with the induction of Th1 biased CD4 positive T cells. And our phase 2 and phase 3 COVE study are underway. I look forward to providing you updates on our progress at future meetings. And then final slide, please. I'd like to end with a thank you and acknowledgement with all of our collaborators. It really has required a village to bring the project to this point, and I want to call out in particular our funding partner, BARDA, the clinical research partnership that we have had with NIAID, as well as the Vaccine Research Center and the COVID-19 Prevention Network. In particular, really strong thanks to our study investigators and all of the study personnel that are working so hard to deliver this study. And then finally, the subjects who agree to participate. They really are the true heroes of this endeavor, and none of this would be possible without their trust and participation. So with that, I'd like to thank you again for the opportunity to share these data, and I'm very happy to take any questions you might have.
Thank you very much, Dr. Miller. Open for discussion.
Hey, this is Kip. I had
Dr. Hunter, please.
Hi. Yes, Paul Hunter. I just have a concern that if convalescent titers were collected later than post-vaccination titers, they might be setting a lower target than we need from a vaccine, if I'm understanding how things work correctly. And so I'm wondering, on average, how long after infection were the convalescent titers collected, especially compared to the post-vaccination titers?
Yeah, thanks for your question. The convalescent titers were collected between 23 and 60 days after the onset of symptoms, and so really represent the range of time points that I presented in the slide deck.
Thank you.
Dr. Paling.
Hi, Kathy Paling. First of all, thank you for presenting an enormous amount of work that has been done and presenting it so clearly. I wanted to give you an opportunity to talk. One of the things that I heard was how we have a predominant of Th1 response and rarely have Th2. Would you mind elaborating on why that's so important?
Yes, thank you for that opportunity. So the Th1 responses are reassuring to us in terms of the potential risk for vaccine-associated enhanced respiratory disease. And the reason for that is in the past with other respiratory viruses, such as respiratory syncytial virus and measles virus, when children have been observed to have enhanced disease after vaccination, when their T cells were examined, they had a preponderance of the Th2 phenotype, so elaborating cytokines such as IL-4 and IL-13. By seeing Th1 both in our animal models as well as in our phase 1 clinical trial, it was helpful to be reassuring to begin clinical development, especially because our clinical development is occurring with relatively rapid enrollment. But I should mention that we're certainly not relying on that information. We have a DSMB for both the phase 2 study, which is supported by Moderna, and a phase 3 study, which is supported by NIAID. And the DSMBs are reviewing the reported safety information throughout the trial and monitoring specifically for vaccine-associated enhanced respiratory disease.
Dr. Kimberlin.
David Kimberlin, AAP Red Book. Are you along the lines of kind of thinking about the inflammation that goes along with natural disease? Are you measuring markers of inflammation in the studies?
So the markers of inflammation had really been measured in the phase 1 study in order to expedite the enrollment and generation of data. The assays in the phase 3 study have been limited to the ELISA and the pseudoneutralization assay. However, detailed information is collected on the patients who ultimately do test positive for COVID-19. And while we're not specifically looking at cytokines, they will be followed closely on a daily basis through telemedicine visits, and hopefully we can then capture the symptomatic data and characterize what we see.
Dr. Talbot.
Yes, can you talk a little bit about how this vaccine will be stored and used, and what kind of requirements for refrigeration or room temperature are required?
Sure. And what I am going to present to you are the stability data that are available now. But as much of the development program continues in parallel, we also continue to generate stability data. And so these conditions will be updated as the stability data allow them to be. So at the time of launch, the vaccine will be shipped and stored up to 6 months at minus 20 degrees. Then the vaccine can be kept at 2 to 8 for 7 days in the refrigerator, and that's really intended for sites that do not have the freezer capacity. The vaccine will be presented in a 10-dose vial without preservative. And so once the vaccine is thawed and the rubber seal is initially punctured, there is a 6-hour window available to administer the remaining doses.
Dr. Freihofer.
Yes, this is a follow-up question about the vaccine-enhanced disease. Earlier in your presentation, when you showed a slide of preclinical data support human clinical trials with mRNA 1273, there was a statement that there was no indication of enhanced respiratory disease after viral challenge, even when the lowest doses of mRNA 1273 are used. Can you explain that a little more thoroughly for me, please? Thank you.
Sure. So in both the mice and the non-human primates, lung pathology was examined after viral challenge. And the viral challenge in the mice was at a dose of 5 times 10 to the 5th. In the non-human primates, it was 7.6 times 10 to the 5th PFUs. And when the animals were sacrificed, no inflammation was seen in the lungs, even down to doses of 0.1 micrograms. So, while we used the 100 micrograms, that statement was meant to reflect the fact that through all of the doses tested in the animal models, we did not see lung pathology.
Dr. Sanchez, my apologies, I skipped you. Dr. Sanchez?
No problem, Roberto. No, that was great. It's very exciting data. I was just wondering, I was wondering actually the issue that's come up now with reinfections in those 3 individuals. And you're monitoring these individuals in the phase 3 study through 24 months. And what are your plans for addressing that in terms of beyond meeting or not meeting the primary endpoint?
So please let me know if I haven't understood the question specifically, but maybe it would help to know that we will follow and capture every case of COVID-19, so not just the first case of COVID-19 in the study conduct. And depending on how many infections we would observe, I mean, we are intending on looking at the request of our DSMB at cases that occur more than once. We also will be looking at antibody persistence at various time points throughout the trial, and we'll be evaluating also by looking at the phase 1 and phase 2 data very carefully to see if there's a certain moment at which it would make sense to evaluate a booster dose.
No, thank you. And I presume you would be doing sequencing of follow-up isolates, or?
So sequenced to the, we don't plan to actually sequence the individual isolates now, but it's definitely an interesting idea, and I'm sure it will now generate some discussion with our partners. We will be testing the individuals for RT-PCR because that's how we will define all of the cases. We are investigating for viral shedding when individuals are actually positive by RT-PCR. So every 3 days or so, for the first 14 days, subjects will either come into the clinic for a nasopharyngeal swab or they'll submit a salivary sample. And so there will be viral shedding samples on which that kind of work could be done.
Thank you.
Dr. Szilagyi?
Yes, Peter Szilagyi. Thank you for this incredible work and very clear presentation. I have a couple of questions about the target population for the phase 3 trial. As you know, there's been a lot of national interest to ensure that the target population is reflective of the U.S. population. And I appreciate the advisory committee and the regular reviews of race and ethnicity. But my question is, are there targets or internal goals for race and ethnicity, and also for age group above 65, so for example, above 75? And could you also define race and ethnicity a little bit more? Are there any goals or targets beyond Black and Hispanic?
So thank you for your question, and I'm going to try to give a more thorough picture of how we are monitoring for race and ethnicity. And this is definitely an evolving picture. I would say it is the predominant topic of the weekly meetings that we have with Operation Warp Speed. So our goal has always been to enroll a study that is representative of the demography in the U.S. And, we know that in the past, it's been difficult, actually, to reach that goal, and very few vaccine trials have been able to do it. So the outreach activities started early on. There are not specific targets at the moment, although they get discussed on a regular basis. And if there is an update to that information, I'll be happy to provide it. African Americans and Hispanic Americans are certainly a focus of the trial, but we are also tracking the enrollment of Asian Americans and of Native Americans, again, because these are very important groups also at risk for COVID-19 disease. And as I said, recognizing that there is so much interest in how the diversity enrollment is progressing, we have this week made a commitment to be transparent about the progress of our enrollment. So we are going to update on our website on a weekly basis this information, including the percentage of non-white enrollment in the trial.
I really appreciate that. And also, well, first of all, I feel that confidence in any vaccine will be related to the representative of the population that has been enrolled in phase 3 trials. So I think this is really, really important. Could you comment on the question about older age groups over 75?
Yes, sorry about that. So there's not a specific stratification for older adults above age 75, not a specific proportion, but I can tell you that we have had patients already enrolled in their 90s. So the hope is at the end there will be a reflective spread of the demography there as well. But so far that has actually not been, the older age cohort in itself has not been a challenge to enroll.
Thank you very much. I recognize the challenge in enrolling minority populations, and I appreciate all the work you're doing.
Well, thank you very much for that. And we fully agree that this is absolutely critical to really assess the value of the vaccine.
Dr. Hayes.
Yes, could you please address any adjuvants that you're using in the multi-dose vial of the current vaccine?
So mRNA 1273 does not contain any adjuvants. It is purely the messenger RNA in the lipid nanoparticle dispersion, and the excipients are limited to tris, sucrose, and acetate.
That's wonderful. So that means that you anticipate that this vaccine this is the vaccine that you will use clinical trials for pregnant women?
Yes, that is the intent. And I should say that recognizing the importance of COVID-19 disease in pregnant women, we are evaluating the potential for further evaluation. I think what we would like to see is a larger safety database in non-pregnant adults before really engaging in that work. But I would mention that in the phase 3 clinical trial, there is an intent to report all pregnancies in the clinical study report. Nonetheless, women are getting pregnancy tests before they're vaccinated, so we anticipate that number to be small. But recognize the importance, and we'll be further evaluating as clinical development progresses.
Thank you.
Dr. Sanchez, your hand is still up. Do you have additional questions?
Oh, I'm sorry, no.
Dr. Freihofer? Dr. Lee?
Thank you so much, Dr. Miller. That was an excellent presentation. Thank you. I had two questions. One, perhaps my main question is really in thinking about the outcomes. You're certainly focused on disease prevention, so capturing patients who are symptomatic with COVID. Is there the possibility to also consider asymptomatic cases? And you had mentioned up to 14 days post-vaccination, but throughout the trial, it may be incredibly valuable for us to understand whether or not your particular vaccine candidate could also play a role in preventing asymptomatic transmission, since we know that that is also of concern here. So that's my first question. And then the second question, is just really if you could clarify. You mentioned and appreciate very much that it's a very Th1 directed phenotype, and the Th2 directed phenotype was rare. But of those who may have, you may have observed a Th2 directed phenotype, is there any comment about those patient characteristics where you observed this finding? For example, recent pregnancy status.
So thank you for those questions. I'll talk first maybe to the phase 3 study design. And I didn't really get into so many details about the COVE study in the interest of time and focusing on the data, but I would be really happy to come back at a subsequent meeting and kind of give a deep dive of all of the various secondary and exploratory objectives that we will evaluate. So what I've reviewed for you are the co-primary efficacy and safety objectives. But in addition to that, there are numerous evaluations of secondary and exploratory efficacy. So in terms of the secondary efficacy, we will look at severe respiratory disease, COVID-19 symptoms according to the long list of potential symptoms that are available on the CDC website, so milder disease, and then also asymptomatic disease. So patients will be monitored throughout the study at various time points, looking at non-spike protein antibody titers. So elevations in those titers and seroconversion in those titers representing the development of an asymptomatic COVID infection. And I would be really happy to go through and review at another point, but we will look at all COVID disease, asymptomatic COVID disease, all-cause mortality, and so forth. So let's maybe plan to talk about that in the future. And then with respect to the Th2, so I really didn't get a chance to show the slides, but there was really, really minimal Th2 directed phenotype. And as that was in the phase 1 study, these were healthy subjects. There really isn't a lot of clinical detail about them, but for us, it's extremely reassuring that the phenotype is really, really heavily biased towards Th1.
Thank you.
Dr. Bernstein.
Thanks for a very clear and wonderful presentation. I just had a question. You mentioned that all comers with regard to SARS-CoV-2 serostatus are included, but baseline serology was still being collected. How will that baseline serology be analyzed with your endpoints?
Thank you for that question. So we will enroll all comers and get the initial baseline serostatus. When it comes time to perform the efficacy analysis, the cases that accrue are really based on its first-time efficacy for that primary analysis. So to be eligible for inclusion in the primary efficacy analysis, subjects should have been baseline serostatus negative. Nonetheless, by including some baseline serostatus positive subjects, we have the ability to conduct some of the sub-analyses and exploratory analyses we were discussing, looking at the history of the subjects who maybe were baseline serostatus positive, evaluating whether cases occur in those subjects. And this is certainly amongst the questions that the DSMB has asked us to look into as we monitor the study.
And what initially are there? What percentage are seropositive?
Difficult to say, I would say at the moment, because in the largest population, which is the phase 3 COVE study, those samples are being accrued and being tested currently. In the phase 1 and phase 2 study, it's been relatively infrequent, I would say at around 1% or so of the study population.
Thank you.
Sure.
Dr. Atmar.
Thank you, and I'll add my thanks for a clear presentation. I actually have a follow-up question to Dr. Bernstein's question about baseline serostatus. And looks like from the phase 1 data, at least by the pseudoneut, you didn't have any seropositives, although by the ELISAs, which may be cross-reactive, I guess, with other human coronaviruses, there were some who had measurable antibody at baseline. Which assay are you going to use to determine baseline serostatus is the first question. And then just a follow-up question to a comment you made during the presentation about making the race ethnicity data available on the website. I took the opportunity to go to the website and see that you have the enrollment numbers through last Friday, but I don't yet see the race ethnicity data. And my question is this going to appear on this week's update, or did I miss something on the website? Thank you.
Yeah, maybe I'll take the second question first, if that's okay.
Sure.
When I last went to the website, I saw the 18% number and the breakdown. So I'll need to investigate what happened there and why you're not able to see it. But the intent is really to provide it and to update it on a weekly basis, given the amount of scrutiny that we're undergoing. And maybe then to the first question, which was around the baseline serostatus and which assay we would use, and looking at the phase 1 data. So yeah, I think there were only 1 or 2 subjects in the phase 1 study that were baseline seropositive, and there were 120 subjects in the trial overall. So it's a relatively small sample size on which to make that assessment. We have seen more baseline serostatus positive individuals in the phase 2 study. At the pre-vaccination time point, we will assess both the binding antibody titers as well as the neutralization antibody titers. And I don't know that we've progressed so much into our statistical development plan to define that, but I mean, presumably positivity with either assay would indicate that you had been exposed previously.
Thank you. And a follow-up question then is, you may not have the data yet, but do you know, are the seroresponses different in those who have baseline antibody, either from the phase 1 or what data you may have from the phase 2 so far?
Yeah. The data from phase 1 are extremely limited, and the phase 2 data are actually currently in their stage of accrual. So subjects have been given the second dose, and now they're in that process of waiting for 1 month post-vaccination to have their immunogenicity assessment. So we should be able to give a better picture on that later in the fall.
Thank you.
Final call for any questions. I see none, so let me thank Dr. Miller again for this excellent presentation, for answering questions. We'll move on to Dr. Kitchen from Pfizer BioNTech, who will talk about their COVID-19 mRNA vaccine clinical development.
Thank you very much. Can you hear me okay?
We can.
Good. Great. Thank you very much to the committee, and thank you for inviting us to present. I work in the Pfizer Vaccine Clinical Research and Development Global Organization, and we, as many of you may be aware, have partnered with the German company BioNTech on development of a COVID-19 mRNA vaccine, or in fact a suite of COVID-19 RNA vaccines. We have been previously working with them in other infectious disease areas, and therefore with the advent of the COVID-19 SARS-CoV-2 pandemic, the platform was right for us to collaborate further. So if you could go to the next slide, please. So what I plan to present is an overview of our clinical development, both phase 1, and I will present some data from our phase 1 study, or part of our study, that directed our decision as to what to move forward with in larger scale development. And then I will present an overview of the phase 2/3 part of the study. So if you look on the right-hand side of this slide, you'll see that we initially started developing in 2 different clinical studies that I'll show on the next slide, 4 different types of mRNA vaccine targeting the SARS-CoV-2 virus. They were encoding either just the receptor binding domain, part of the spike antigen, or the whole spike, again, the P2S pre-fusion stabilized form. The 4 different candidates based on either unmodified RNA, modified RNA, or self-amplifying RNA. And we have focused the attention of our large-scale development and what I will talk about further moving forwards on our 2 modified RNA candidates given as a 2-dose schedule separated by 3 weeks. The so-called B1 candidate, which encodes for the RBD only, and the B2, which encodes for the P2 mutated full spike protein. Next slide, please. So I mentioned that there are 2 ongoing clinical studies assessing the safety, tolerability, immunogenicity, and ultimately moving forwards now efficacy of these mRNA vaccines. The initial study in the US, shown there on the left, started off as a phase 1/2 study, but has now formally expanded into a phase 3 efficacy evaluation that I will explain in a bit more detail later on in the presentation. And then in Europe, in Germany, a study is being conducted whereby all of those 4 different types of vaccine are being evaluated in different doses. And the importance for that study, particularly for this presentation and for our decision to move forward, is that T cells were collected and analyzed in that study as opposed to in the US study. So in the phase 1 part of the US study, we enrolled 15 healthy participants per dose level, of whom 12 received the active vaccine and 3 received placebo. And we enrolled them in 2 different age groups, either 18 to 55 or 65 to 85. These subjects were healthy and were pre-screened for absence of antibodies to SARS-CoV-2 or positive PCR on a nasal swab at the time of vaccination. We initially planned to evaluate ascending dose levels of 10, 30, and 100 micrograms. But for the 100 microgram dose level for the B1 candidate, we saw a tolerability profile that we did not think fitted with a vaccine that was going to be implemented at a population level, and we therefore turned our attention to lower dose levels in the range of 10 to 30 micrograms. And as I said before, that's administered as 2 doses at day 1 and day 21. And just to note, because of that reactogenicity finding for the 100 microgram group, they did not receive a second dose. We have also made comparisons informally with a human convalescent serum panel from 38 human convalescent sera. You can see in the box there from subjects over a broad age range of 18 to 83, which were collected at least 14 days after a diagnosis, which was PCR confirmed, and once subjects were asymptomatic. The majority, 35 of the 38, were symptomatic infections, and 1 has been hospitalized. And if you wish to view these data further, there are a number of publications cited there at the bottom, either formally published, having been peer-reviewed, or in pre-publication format on medRxiv. You can go to the next slide, please. So this is an overview of the study that's currently running in the United States that I mentioned briefly on the previous slide. So on the left-hand side, I'll focus my attention on to start with, and the right-hand side that's grayed out, I'll come back to later on when we talk about the phase 2/3 component. So in the phase 1 part of the study, we enrolled 195 subjects, as I said, 15 per group in those 2 age groups that you can see shown there in the table at the various different dose levels. And as is usual, in our early phase vaccine studies, we describe the safety and the tolerability profile via participant-reported outcomes solicited via electronic diary, local reactions, systemic events including fever and also use of analgesics or antipyretics, as well as adverse events, and we collect all adverse events for 1 month after the second dose and serious adverse events for 6 months after the second dose. And in this early part of the study, we also collected standard hematology and biochemistry samples. In terms of immunogenicity, we've assessed this in a number of different ways, focusing our attention on neutralizing titers, analyzed using a true viral neutralization assay, as well as looking at binding antibodies against either the S1 or the RBD. However, what I'll present here will just be the neutralizing titers, as we feel that those are likely to give the best representation of potential future performance of the vaccines. You could go to the next slide, please. So in summary, and I'll show you the data that supported this, of the B1 versus the B2, so the vaccine encoding the RBD versus the vaccine encoding the full spike protein. We selected the B2, so the full-length spike encoded vaccine, at the highest of the 3 refined dose levels that we looked at, the 30 microgram dose, and that is our focus for the late-stage phase 2 and phase 3 development. And we used a number of different criteria to determine how best to choose both the candidate and the dose level based on obviously acceptable safety and reactogenicity of paramount for a vaccine potentially for wide-scale global use. We were focusing our attention from an immunogenicity perspective, as I said, on neutralizing titers, and we did use the human convalescent serum panel as an informal reference. And we also wanted to see a strong Th1 type T cell response, both for CD4 and CD8 positive T cells. And both candidates looked potentially strong, but overall the data favored the choice of the B2 candidate, the one encoding the full-length spike protein, based upon, and I'll show you some of these data on the following slides, the reactogenicity profile for the B2 being more favorable than the B1 in both the younger and the older adults, a trend towards stronger CD8 T cell responses with the B2, and earlier clearance of SARS-CoV-2 RNA in immunized and subsequently challenged rhesus macaques. So based on all of these data, it is the B2 candidate BNT162b2 at the 30 microgram dose level that we're studying in the large part of the study. Next slide, please. Okay, so let me show you some reactogenicity data from the phase 1 part, if you could go to the next slide. So first of all, what I'm showing here for the B2 is the local reactogenicity profile in phase 1. So just to orientate you, there's going to be a couple of slides with a slightly similar layout. So in the top half of the slide, you have the data after dose 1. So this is, just as a reminder, this is participant-reported outcomes in an electronic diary on the evening of each of the 7 days after vaccination, and the data presented cumulatively. So dose 1 in the top half of the slide and dose 2 in the bottom half of the slide. And then on the x-axis, you can see the various symptoms that were solicited for and the various dose levels, the 10, 20, and the 30 microgram dose levels, and then the P standing for placebo. The left-hand side of each graph shows the 3 local reactions solicited for in the younger age group, the 18 to 55-year-olds, and on the right-hand side, exactly the same data presentation, but for the older age groups. And then each bar is a stacked cumulative bar chart with the light green showing the mild symptoms, and just I should have said earlier that these local reactions and systemic events were all graded by participants according to the FDA grading scale. Standard FDA grading scale. So the light green is for mild, the yellow for moderate, and then the darker teal for severe. So you can see that across all 3 dose levels at both doses and in both age groups, Pain at the injection site was the most common local reaction, perhaps with an indication, at least in the younger age group, of bearing in mind the small numbers, of course, increasing with dose, but less common in the older subjects than in the younger subjects. And you will see that redness and swelling, whilst pain is a predominant symptom, redness and swelling were relatively uncommon. You go to the next slide. So let's turn from local reactogenicity to systemic reactogenicity. So again, we've got a similar layout here, but now because we solicit for more systemic events than we do local reactions, what I'm showing on this slide is, first of all, is the younger age group, the 18 to 55-year-olds, but again, dose 1 on the top panel and dose 2 on the bottom panel. Again, this is just for the B2 candidate. So you can see, particularly if we focus our attention on the more objective symptoms, so particularly fever, for example, you can see that in the younger age group, after dose 1, there were just 2 subjects reported fever, 1 mild and 1 moderate after dose 1, and 3 after dose 2, 1 in the 20 microgram group and 2 in the 13 microgram group. In terms of chills, again, perhaps more objective than things like fatigue and headache, and that's borne out when you look at the rates in the placebo group compared also to the active vaccine groups. You can see, again, there does appear to be an increased frequency with increasing dose, but again, very tolerable. These reactions tend to occur on either day 1 or day 2 after vaccination, and are short-lived, resolving within 1 to 2 days. So that's for the younger age group, the 18 to 55-year-olds. And if you go to the next slide, I'm sorry. This is exactly the same layout for the older age group, the 65 to 85-year-olds. And I think you perhaps saw when the slide transitioned there that the overall height of the bars on this graph appear lower than on the previous graphs, indicating better tolerability profile amongst the older subjects compared to the younger subjects. And in particular, if you look at fever, for example, in all of the dose levels, there was only a single report of fever in 1 subject in the 30 microgram dose group, mild, so 38 to 38.5 degrees centigrade after the second dose. So I've not presented the B1 data here, but this did show these tolerability data were preferential compared to these B2 data were preferential compared to the B1, and that was one of the key factors that helped us choose the B2 encoding the full spike over the B1 encoding the RBD only. If you could go to the next slide. The other thing to say is that there were no, in the phase 1 part of the study, there were no serious adverse events reported by any subjects. So in terms of immunogenicity, I've got one graph to show on immunogenicity, and again, let me orientate you. It's quite busy. So here, the majority of the data are for the B2 in the teal colours, but I also included the B1 30 microgram dose level just as a comparator and as a reference. So what you can see is the 18 to 55-year-old age groups on the left and the 65 to 85 on the right. These are 50% serum neutralizing titers in a true viral neutralization assay. The placebo subjects are grouped together on the left in the gray. Then each of the available time points for the various dose levels are shown along the x-axis. So vaccinations given 3 weeks apart, so at day 0 and day 21, and then the 7-day post-dose 2 sample taken at day 28. On the extreme right-hand side are the results from the human convalescent serum panel that I referred to earlier. The heights of the bars and the numbers shown above them represent the geometric mean titer, and each individual point is an individual subject. And these are all available immunogenicity data, and I'll explain why that's notable in a moment. So what I think you can see is that comparing the 10 with the 20 and the 30 microgram dose levels in the younger subjects, there is an increase in the geometric mean titer after the second dose, particularly at day 28. We do see binding antibody at day 21, but the neutralizing antibody really picks up after the boost with the second dose. And just to point out my comment about these being all available immunogenicity data for the 30 microgram dose level that we actually selected, you will see if you've got eagle eyes that for the day 28, there is a teal triangle right down there on the baseline of the axis. That was in fact a subject who had their blood sample taken out of the protocol window. That was only 2 days after the second dose. So when that subject is excluded, the geometric mean titer is at a similar level to seen after the 20 microgram. And you can see if you compare the 30 microgram dose levels that the B2 and the B1 show rather similar levels of neutralizing antibody titers. Again, so therefore, our selection of the B2 on the basis of similar immunogenicity but better reactogenicity is what drove that. The other thing also notable on the slide is that if you compare the younger age group to the older age group for the same dose level, both for the B1 and the B2, you can see that the geometric mean titers are lower in the older subjects than in the younger subjects, as might be expected. However, what's key is that if you compare in all of the groups after they've received the second dose, you can see that the GMTs are significantly higher than that observed in the human convalescent serum panel. Of course, it's difficult to know at this point in time what the true meaning of human convalescent serum neutralizing titers are. However, I think they represent the best benchmark that we have at this point in time, and to help guide our selection as to what to take into further development. If you go to the next slide, please. And then finally, in terms of immunogenicity, we did not collect T cells in the US study, but we did collect T cells in the German study. I've just got one slide to show on those. If you could go to the next one. What we see is, and this I just want to make clear, this is for the B1 candidate. The data for the B2 are still under preparation. However, what I can say is that the data for B2 look at least as good as the B1 and show a similar pattern in terms of inducing strong both CD4 and CD8 positive T cell responses. Again, with a Th1 dominance. So if you look there, you can see the increase between the pre and the post. So that the pre is before the first dose. The post is on day 29, so a week after the second dose. You can see nice rises in interferon gamma and IL-2, and very little response for IL-4. Next slide, please. You go to the next slide. Thank you. So our phase 1 data gave us encouragement that from both safety and an immunogenicity perspective, advancing BNT162b2 encoding the full-length spike antigen looked like a good candidate from a reactogenicity perspective, where the reactogenicity we observed was lower after the first vaccination compared to the second, was lower in younger than older participants. Sorry, lower in older participants. That's the wrong way around. Apologies. And the profile appears at least as good as approved adult vaccines, and therefore should be supportive of a widespread immunization program. In terms of immunogenicity, we saw strong neutralizing antibody responses from 7 days after the second dose. We are still collecting data. These subjects will be enrolled, will be included for 2 years after their second dose, and we are still collecting and analysing data from time points beyond that, obviously, but we don't have those data yet available to share. And finally, we saw, again, I think important, both strong CD4 and CD8 T-cell responses with a Th1 dominance. So we took, if you go to the next slide, we therefore elected to take the BNT162b2 candidate into phase 2/3 development, and then the next slide, at the 30 microgram dose level. So if we now focus on the right-hand side for this part of the study, we have, so the phase 2 component represents the first 360 subjects enrolled in the overall larger phase 2/3. So 180 per age group and per vaccine. So they are randomized 1-to-1, unlike the first part of the study where the randomization was 4-to-1. In this part of the study, where our primary endpoint for the phase 3 part is efficacy, we are randomizing 1-to-1 active to placebo, aiming to enroll an overall target of approximately 30,000 subjects. And you can see the age split there. So we're targeting approximately 60% of the subjects to be in the 18 to 55-year-old age group, and about approximately 40% to be in the 56 to 85-year-old age groups. The primary objectives in this part of the study are for the phase 2 part to further define the safety profile and the immune responses to the BNT162b2 in those first 360 phase 2 participants. And then for the large phase 3 portion, the efficacy objective is to evaluate the efficacy of the vaccine against confirmed COVID-19, firstly in those without evidence of infection before vaccination. So we take a baseline serum sample, which we evaluate for a non-vaccine antigen, and nasal swabs for presence of SARS-CoV-2 by PCR. Again, we don't exclude subjects based on either of those parameters. They are included, but for the first part of this primary objective, they would be not included in the analysis because the first part is only in those without evidence of infection before vaccination. And then the second part of the objective is to include all comers, both those with and without evidence. And again, that's important when considering implementing a vaccination program at a population level where clearly there will be vaccinees who will be naive, but also those who have been previously exposed either knowingly or unknowingly. Of course, the safety profile is paramount for this type of vaccine. Again, we will be assessing local and systemic reactogenicity via electronic diary in a subset of participants, and adverse events in a similar way to the phase 1 part of the studies, including all adverse events up to 1 month after the last dose, and serious adverse events up to 6 months after the last dose. And we do have additional secondary and exploratory objectives to look at severe disease, looking at using the broader CDC case definition, and looking at SARS-CoV-2 infection. Next slide, please. So in a simple schematic in terms of how we surveil for efficacy, so this study started on the 27th of July. I'm pleased to report that the enrollment is already more than 50% accrued. So subjects will receive 2 doses of vaccine in the same way as in the phase 1 part, separated by 3 weeks. And as well as capturing reactogenicity and adverse events, we will be doing intensive surveillance for potential COVID-19. If a subject experiences a symptom that could potentially represent COVID-19, they are to contact their investigational site, and that triggers either a telehealth or an in-person visit and a nasal swab. And then we will be defining our cases based upon both the presence of one or more symptoms as well as a positive SARS-CoV-2 PCR positive. And as I said before, we'll be looking at efficacy both in first of all in those without evidence of infection, and then next in the total population. We plan to follow subjects for up to 2 years after the second dose with regular follow-up visits. We too are focused on enrolling a diverse population. What I should have said also is that this is an international study, including the United States, Latin America, Europe, and South Africa. The majority of subjects will come from the United States. Our current enrollment shows that we have approximately 19% of subjects enrolled either Black or Hispanic, and 4% Asian, and we are also working with sites to enroll Native Americans also. And obviously in Latin America and in the other regions, there will be increased diversity also. And with that, I think that is my final slide. If you go to the next one, I'm now going to hand over to, I'll be happy to take questions afterwards and have other colleagues on the line who can also answer questions if you have them. But I'm now going to hand over to Brian Gleason from our global suppliers to talk about the storage and handling of the vaccine.
Good morning, good afternoon. Thank you. Brian Gleason, I'm here representing Pfizer Global Manufacturing Supply, and I am the Pfizer launch lead for the vaccine. So I'd like to thank ACIP for the opportunity to present today. Next slide, please. So the first slide, I'd like to give you an overview of the product packaging for our supply for this vaccine. The first primary packaging on the left side of the slide is going to be filled, formulated drug product will be filled into 2 ml type 1 glass preservative-free multi-dose vials. Each multi-dose vial has 5 doses, and each multi-dose vial has 0.45 milliliters of frozen liquid drug product with point-of-use dilution required. The second step along is the secondary packaging, and that will be a single pack into a tray. Each tray will have 195 2 ml vials, which gives us a total of 975 doses based on the 5-dose MTB. Each tray is approximately in inches, about 10 inches square, or 230 millimeters square. After secondary packaging, it passes on to thermal shipping into a tertiary container. And here we have designed an innovative, flexible solution to ensure the maintenance and tracking of our required cold chain conditions. Each thermal shipper will utilize real-time temperature monitoring devices, including GPS tracking technology, to allow for 24/7 in-transit control, enhance security, and also allow for any mitigating actions on temperature deviations if required. Each shipper can take up to 1 tray, up to 5 trays. So you can fit 1 or up to 5. So 1, 2, 3, 4, 5 trays into a thermal shipper. It'll be submerged in dry ice and packed up for shipping. The total fully loaded weight is approximately 35 kgs, or about 70 pounds. Next slide, please. Okay, so when the thermal shipper arrives at the point of vaccination, there are a number of options. On the left side, the first option is to remove the vaccine tray or trays from the thermal shipper and store in a ultra-low temperature freezer at -70 plus or minus 10 C, where it can be stored as a frozen liquid for up to 6 months. There are many different sizes and designs of ULT freezers available. Smaller underbench type pharma grade freezers can hold typically up to about 30,000 doses. And the more familiar, larger upright design freezers can store well over 200,000 doses. So flexibility on freezers is there in the marketplace. Long-term stability studies will continue under protocol to inform drug product shelf life at -70. Second option in the middle of the slide, you can utilize the thermal shipper as for extended storage. So each shipper is validated to keep the vaccine payload at ultra-low temperatures for up to 10 days. And on receipt at the vaccination center, after opening, we recommend that the thermal shipper should be replenished with dry ice. So there's a dry ice replenishment needed within 24 hours. Thermal shipper can be replenished every 5 days, and you can extend the storage every 5 days with a replenishment. So that's the keep it for an extended period. If there's difficulties in sourcing dry ice services locally, Pfizer has strategic suppliers, and we have a reference list which we will share in due course. The recommendation for the thermal shipper is not to open more than twice per day, obviously to elongate the cold storage, and after opening, also recommends to close within 1 minute. On the right side, the third option is to remove the vaccine from the thermal shipper and place it into a refrigerator at 2 to 8 degrees C, where based on current stability data, it can be stored for up to 24 hours. We have ongoing stability studies which will continue to characterize our drug product stability. And over time, the expectation is the new data from these studies will help to optimize or allowable storage temperatures at 2 to 8. Post-dilution, the in-use period is expected 6 hours at 2 to 30 degrees C. Okay, that's the last slide. Thank you for your time.
Very good. Thank you both for those presentations. Dr. Cohn wanted to say a couple of words first, and then we'll open it up to questions.
Thank you so much for all of that really important information that you just shared for both products on vaccine storage and handling. I just want to let all of the listeners know to not start going online right now and purchasing freezers. We are working on solutions through our distribution and administration planning for these very complex storage and handling requirements at this time. Additionally, I think hopefully Pfizer can comment on sort of a timeline on additional studies on stability that may shift these plans. But I want to let everyone know that as soon as we're able, we will start sharing information about how CDC and HHS through Operation Warp Speed are going to be supporting supporting programs at maintaining this vaccine at this temperature. Over.
Thank you, Dr. Cohn. So I will take Chair's prerogative and ask first a couple of questions. First of all, the timing of your signs and symptoms for adverse reactions, do you have a window for that? Because that's going to cause some problems because they mimic signs and symptoms of COVID and may lead to excessive COVID testing in this population and in vaccinees, excuse me. And then second of all, what are your plans for measuring antibody persistence over time?
Thank you very much for the question. So we do provide some guidance to our investigators about recording potential COVID symptoms in the first 7 days after vaccination, since we rightly point out there is a potential overlap between some of the more common symptoms and the reactogenicity. So we don't anticipate an over-testing for COVID during that period based upon that guidance. In terms of persistence of immunogenicity, so the study is currently planned to follow subjects for up to 24 months after they've been vaccinated, and we will have bleeds across that whole period. So we will have, at least for now, those, and obviously these are going to be the first subjects vaccinated in hopefully what will be an ongoing program.
Thank you. Dr. Lee, please.
Thank you very much. Two questions. First is, and actually this applies perhaps for all of the trials we'll be seeing, are there ways for us to capture the full benefits of vaccination? So I recognize that respiratory disease or some of the systemic symptoms that are presented are sort of classic COVID presentations. With regard to the potential to capture other disease manifestations secondary to COVID infection, and particularly as we in the future get to trials in children, I think it might be helpful to widen and capture that. Perhaps it's in your secondary outcomes. And again, I'll emphasize that asymptomatic infection and thinking more about the response earlier from captured via PCR versus serology. I actually feel like with serology and asymptomatic infection, we sometimes are not seeing as strong a response as we are with symptomatic infection. And also, in order to really think about herd immunity in the way that we would like to, such as comparing polysaccharide vaccine to conjugate vaccine in kids and the differential impact it has on the population, it would be extremely helpful for us to understand the potential for each of the vaccine candidates to have that impact. And then I have one more question, but I'll pause there for a second.
Thank you. So I think, sorry, I'm just trying to assemble what you said. So I think a number of those things that you've mentioned are probably going to have to be studied in further studies, and in fact, perhaps even in following assuming efficacy and safety are demonstrated in implementation studies. For example, looking at herd immunity and that type of thing. For our part, we have tried to keep, bearing in mind the number of subjects needed to be enrolled in the study and the urgency. We've tried to keep the study as pragmatic as possible in the first instance, rather than overloading it with a lot of additional procedures that might make the implementation of the study difficult. But I think those are things that we can come back to look at in future studies. And the other thing to say is that you mentioned pediatrics there. We are also we're in dialogue with regulators both in the US obviously and also elsewhere about plans for pediatric studies that we as well as studies in pregnant women. And in other potential groups that we would be looking to implement as early as we can.
Thank you, Dr. Kitchen. One more question. I just wanted to clarify. So with regard to the safety follow-up in the phase 3 studies, I saw on one slide that for severe adverse events, it's potentially up to 6 months. And also on another slide that you're following patients up out to 2 years. With regard to efficacy and post-vaccination infection. Is it also possible to clarify whether or not you'll extend safety monitoring out to 2 years, at least for the serious adverse events?
So yes, you're right. Well, you're right that it's surveillance for serious rather than severe. Serious adverse events, and that's an important distinction, up to 6 months after the second dose. That's if you like the active surveillance. It doesn't preclude reporting of adverse events beyond that, and we have some experience of long-term follow-up vaccine studies where investigators continue to report adverse events during that period. So that's the current plan for reporting of all serious adverse events. I think beyond that would then depend upon, if any adverse events of interest or signals were observed that we needed to focus on additional safety surveillance beyond that. But investigators are at liberty to report adverse events at any time during the trial.
Thank you. And maybe just as a comment, not specific for perhaps for all trials, for us to consider standardizing the safety monitoring post-vaccination in the phase 3 study, just because I think, each of the vaccine candidates may potentially have unique profiles, but also just thinking about as we're assembling the data for vaccine safety and reviewing it at ACIP, I think it would be helpful for us to have a standard approach to that. Again, not directed, solely at you, but just as a general statement. Thank you.
You're welcome.
Dr. Szilagyi.
Yeah, thank you very much for your presentation. I did have one question about pediatric population, which you addressed. But the other one, for some reason, I'm unclear about this. Are the 18 to 85-year-olds healthy individuals? Because I know some centers are enrolling healthy individuals. And what is the representativeness in terms of underlying high-risk conditions?
Yeah, so thank you for the question. And I should have maybe said more about the inclusion criteria. So in terms of underlying illness, we are including a healthy population currently between the ages of 18 and 85, but are also in dialogue to potentially extending that range. But when we say healthy, that permits subjects with, or potential participants with, sorry, my telephone, I don't know if you could hear that, my telephone started ringing on the same speaker as I'm trying to speak, sorry. That's put me off slightly, what was I saying? Yes, so healthy participants can include subjects with chronic medical conditions, provided they are considered stable, and we define stable as meaning no change in therapy or significant intervention within the past 6 weeks. So we do already have significant numbers of, and that's the same across all sites. Just to be clear, I'm not sure if you were suggesting that some sites are recruiting different types of subjects than others. Those criteria in our study apply across all of the locations. So we already can see from the baseline medical history data that we've started to accrue that there are significant numbers of people with underlying medical conditions. And as we accrue more, we'll be able to better represent what that population looks like. But our intention, obviously within the constraints of a phase 3 efficacy study, are to recruit a population that is as representative in all ways as possible.
Okay, that's helpful. Specifically, I was thinking about hypertension, diabetes, obesity, chronic lung diseases.
Well, I can tell you certainly that hypertension and obesity and diabetes are at the top of the list of the, we've got significant numbers of subjects with those conditions already enrolled.
Thank you.
Dr. Paling.
Hi, Kathy Paling. Dr. Kitchen, thank you for this great presentation. I wanted to ask for clarification. I saw that under phase 1 and 2, you said there was a predominance of Th1 response, and you showed data really for the B1, and we're going to be moving forward to B2. Could you share what we've seen about with the Th2 response? And then second is, would you clarify if in the phase 3 study, the Th response is going to be included, aligning to what Dr. Lee had talked about, making sure we're comparing similar things across studies?
Yeah, so the operational complexity of collecting samples for T-cell analysis is quite substantial. So typically we will only collect those in relatively small, very controlled settings. So in this large phase 3 study that we're now conducting, we do not plan to collect T-cell samples. However, I think the nature of those analyses means that the analyses that we're able to perform in the early phase 1 studies give a good representation of what the T-cell responses would look like. In terms of so I did present those data for the B1 candidate only. So what I didn't say in the presentation was that the subjects that received the B1 were in fact the first ones enrolled into our study. So therefore the data for them comes available sooner than for the B2 who were recruited a few weeks later. So for the B2 data, what I can say is that at this stage, the preliminary data looks at least as good as for the B1, and we hope to be publishing that in very short order, probably within the next week or so, at least on a preprint server.
Ms. Mata.
Yes, thank you. This has been great to hear these presentations. In the slide where you talk about the efficacy schema, which has now just started, in the discussion for active surveillance, you're saying that you're either going to do telehealth or in-person visit and nasal swab. What's the criteria? Like, what? Why aren't you doing nasal swab for symptomatics in all the subjects, or maybe you are? Can you just explain that a little bit further?
Yeah, sure. Yeah, and the short answer is yes, we are. Maybe I wasn't clear enough. So the way that it works is that if somebody experiences one of the trigger symptoms, which are the typically recognised symptoms for COVID-19, they are advised to contact their investigational site by telephone in the first instance, and if it is confirmed that they do indeed have one of those symptoms, they are asked to either when they're first enrolled, they're given a swab kit to take away with them. Then, depending upon their circumstances, the country in which they are enrolled, and the site's circumstances, they can either perform a self-nasal swab and ship that directly to us, or they can attend the site and have an in-person visit and have the swab performed there. But either way, anybody that has confirmed, and when I say confirmed, I mean by the investigational site staff, a confirmed symptom that could represent COVID-19, they are all intended to have a swab performed. I hope that clarifies.
Dr. Freihofer.
Sandra Freihofer, American Medical Association. On your phase 2-3 efficacy schema, which started on July 27, 2020, you said that you are collecting diversity information on the participants, and right now 19% are Black or Hispanic and 4% are Asian. Obviously, from all the questions you've heard, this is very important to so many people. We have a population to cover. This is important not only to trust, but with so many different vaccines that are under investigation with possibly differing profiles, this sort of diversity information on the participants would be so helpful. Are there any plans to include that information specifically on your website?
So I don't know the answer to that. I don't know if one of my colleagues on the line is able to speak to that more. All what I can tell you is that it is like you. It is something that we are very cognizant of, and particularly with the the pattern of COVID and how it disproportionately affects minority groups. That's something we are actively seeking working with our partner sites to put in place tools to help with outreach into the population to try and, because as was said earlier, it's not straightforward to enroll minority groups in large numbers into clinical trials in general, and I think we've made good headway so far, but are always seeking to do better.
Dr. Kimmerlin.
Another of the characteristics of this disease is inflammation throughout the body. Are you all measuring any measures or markers of inflammation, even something as crude as SED rate or CRP?
We are not measuring those in our study, no. We do capture, if parameters are captured as part of someone's medical care, those sorts of parameters, we will catch those, but we don't have them performed under the study protocol.
Dr. Freihofer, do you have additional questions?
Yes, Sandra Freihofer for AMA. One more question. You indicated on your slide showing phase 1 showing reactogenicity that there was an error on your slide, that there was actually less reactogenicity in older versus younger participants. Does this any way make you concerned that perhaps the vaccine may not work as well in older adults? And maybe there's something you've missed. But that was an error on your slide. Is that correct?
So you'll see from the graphs that I showed that the reactogenicity is lower in the older subjects than in the younger subjects, and I also showed that the neutralizing antibody titers were lower in the older than the younger subjects, but were at least as high as the human convalescent sera. So ultimately, the efficacy within the study as a whole will be how we can assess the vaccine's effect overall and in different parts of the population. But until we have those data, we can't really speculate about that.
Dr. Cohen, question?
Thanks, all. Can you please go into a little bit more detail about the breakdown of the age groups in terms of the younger population and the older population in the immunogenicity data and safety data from the phase 1 trial? And also, specifically in the older group, what is the proportion of patients over 70 versus between, I think it was 56 and 70?
So, was that question to Pfizer? Yeah, so just to be clear, so we enrolled in the phase 1, we enrolled 2 age groups. So one was 18 to 55, and then the other was 65 to 85. So in the phase 1 part, we didn't. Sorry, there's a.
Apologies. I think I meant for the 65 to 85.
Right.
How many? It was not clear what the ends were for each of those 2 different groups from the total number of enrolled, and what the range was within the older age group. Not the range, but the proportion of patients on the older end of that.
Right. So just to be clear, yeah. So each group that received the active vaccine comprised 12 subjects at each dose level. I don't have the exact. So bear in mind that's a relatively small number in terms of looking at the overall distribution. But we did have representation across that whole age range from 65 through 85.
We have a question from Dr. Maldonado.
Yeah, sorry, I couldn't raise my hand. Thank you for this presentation. So I understood from the previous speaker that they weren't going to be collecting samples for genotyping, but it seems to me that if you have a breakthrough infection or an infection symptoms and you're collecting swabs, would you be planning on collecting, since you're collecting swabs, wouldn't you want to genotype those to see what the sequences look like compared to the circulating strains?
So we don't currently plan to do that. We currently plan to use those swabs to assess by PCR whether there's presence of SARS-CoV-2 genomes or not. That's certainly something we could think about looking at in the future.
Dr. Paling, I still see your hand up. Did you have another question?
Yes, I wanted to follow up real quickly. And if I understood correctly, the B2 Th1 Th2 data is being put together for publication soon, and I wanted to ask if there were any characteristics or in those that developed a P2 Th2 response.
Dr. Gruber, is that something you're able to talk to?
Yes, hi, this is Bill Gruber. I head Vaccine Clinical Research and Development. I make sure I understand the question. Could you state the question again, please?
Yes, hi, Bill. Kathy Paling. So the question is that when you're looking at the Th1, Th2 responses with the B2 vaccine, were there any, we understand there's a predominance of Th1 response. Of those that had a Th2 response, were there any characteristics that came to light?
Yeah, I would say again that the shift was so dramatic in terms of Th1, there actually was very little to sort of discern where you got actually, for instance, IL-4 response. So I would say there was nothing distinguishing in that group. As Nick has indicated, we continue to look at that data and are preparing it for publication. I will say, and we've commented on this in a press release, and I think the data that will be forthcoming will be available for people to review. As you know, the very nature of the platform lends itself to a Th1 response, and that's part of the reason it was attractive to begin with. I think the data is supporting that, and in fact, by virtue of having the larger antigen to be able to, or a set of antigens to be able to stimulate immune response by using the full-length spike protein, we have the potential, and I think are beginning to see some data to suggest that, in fact, there is greater repertoire of CD4 and CD8 response because you've got more epitopes to respond to. It wasn't clear, obviously, from the very start, where most of the attention would be directed, and that was part of the reason for looking at receptor binding domain versus looking at the full-length construct. But we're seeing, for instance, CMI responses against the full length, both the upper end of the peptide, the N-terminal end, as well as the carboxy terminal end, when we separate it out. And again, that data will be forthcoming. But I would say that the response is so robust, there doesn't seem anything to dissect out in terms of a Th2 response because there's so little there.
Thank you. We'll take two more questions. First from Dr. Quach and then from Dr. Messonnier. Dr. Quach.
Yes, thank you very much. I was just wondering, given the decreased sensitivity of the nasal swab compared to the NP swab, how are you going to handle these differences between people who are coming in to get swabbed at the vaccine center versus those that were going to do self-swab at home?
So just to be clear, the same swab is used for either procedure. So it's a nasal swab as opposed to an NP swab. But it's just whether it's the subject that performs it themselves or whether the site staff perform it.
And so you're not worried at all about the decreased sensitivity of the test compared to the NP?
I mean, I think from some of the data that I've seen, if the amount of virus that is present when people have true symptoms caused by COVID, that's not something I'm overly concerned about, no.
Okay, thank you.
Dr. Messonnier.
Yeah, thank you. I want to start by thanking Pfizer and Moderna and all of our pharmaceutical company partners for their work to try to rapidly develop these vaccines and fulfill a clear public health crisis need. And I understand that, our need to move quickly sometimes means that we have to deal with the realities of imperfections. So in that context, I do want to ask, as I'm sure Pfizer realizes, that the complexities of this plan for vaccine storage and handling will have major impact in our ability to efficiently deliver the vaccine. So in that context, could you provide any more detail as to when you will have more information about storage and handling conditions that are less stringent, and when you will have more information about the possibility of shipping it in boxes that are less that have less quantity. In other words, smaller boxes, which would also provide more flexibility in implementation. Thank you.
Yeah, thank you for the question. Brian, are you still on?
Yes.
Yes, thank you.
Maybe I'll take the last one first. Yeah, so as we said, for the pandemic supply, the current minimum shipment quantity is the 195 pizza box. We're investigating the viability of a less than a 195 count SKU, so a smaller box. We're not able to confirm this time whether that's what the feasible date for that would be, but we're currently looking into that. So again, it's something we can come back to you on.
All right. Thank you very much, Dr. Kitchen, and thank your colleagues for the presentation and answering the questions. For all, we are now going to go to lunch because we want to stay on time. And you're all on different time zones. We will meet again in 20 minutes at 1:30 the hour. Thank you all very much.